leave the remaining 10 minutes for Q&A. So without further ado, I will hand over to Mark for him to start his exciting seminar. Over to you, Mark.
Good morning, everybody, or at least good morning here in the United States, or early morning. I want to thank you for the opportunity to present to this esteemed group of scientists in Southeast Asia and in the European Union, or UK in particular. So today I'm going to bring up a screen about our cross-protective adjuvanted self-assembly nanoparticle H5 2.3.4.4b Astrakhan subunit vaccine candidate, just as an example of the power, the speed, the low cost, and the flexible manufacturing process of what we call the C1 vaccine technology. And I'm going to go to the full slideshow, I guess. And there we go. Can you see my screen, Tuck, and the group?
Yes, yeah.
Okay, great. Just need to move that up. And it's not, there we go. So the C1 technology has been developed over 3 decades, initially for the industrial applications of making industrial enzymes for animal feed, food, textiles, pulp and paper, very, very large scale production. We were used to run at 500,000 liter scale. So I think you'll realize here we're scaling down. And this is a global solution in terms of being able to make the quantities of antigens for vaccines, also for monoclonal antibodies and other therapeutic proteins. And the technology has been vetted and used by some of the largest companies on the planet, including DuPont and BASF and Shell Oil and others. It has US FDA GRAS status from the industrial standpoint, which basically means the pedigree of the cell line, which you can see on the left, the C1 cells actually is a better starting point than E. coli, which you have endotoxins, or insect cells like baculovirus, where you have viruses, or CHO cells that need to be removed. But the C1 cells, we do not have to remove those because they don't exist in the first place. So you save 2 steps in the downstream processing from viral, for example, viral clearance, and you can do it faster. The technology has also been used in the biopharmaceutical industry, where we've expressed dozens, if not close to 100 now, different antigen proteins and antibodies and therapeutic proteins for our cells, big pharma, small biotech, academia. And we work in collaboration with academia, industry, governmental agencies, where we take a gene sequence and turn them into proteins. And when they then create stable cell lines, which we provide the protein back for you to test and evaluate in your preclinical studies. And then we can out-license you the technology platform to bring it in, where you get all the genetic tools to put DNA in and out. We've knocked out 15 of the different proteases in the background, and we also have a humanized glycoengineering strain as well. One of the unique things also about C1, you look on the top left, is the morphology of the cell. This filamentous fungi doesn't sporulate in manufacturing, and it's changed the morphology, so that led to a reduction in viscosity from 10,000 centipoise to less than 10 at the end of the fermentation. So now I'm going to start getting into some of the advantages of the platform itself. It can be used for both animal and human health. So C1 is a very rapid way to produce technology and stable cell lines. You can see here we have precise and effective genetic engineering technologies. We can do site-specific integration, we can do random transformation, or we can also use CRISPR and hit multiple sites at one time. We can put single copy integrations, double copy integrations in a defined complex low viscosity media that's tailored for fermentation, pH, and temperature. And you can see in the bottom left, we produced a MAB at 24 grams per liter in 7 days. And on the far right, you can look at what we've done here with what this happens to be an animal antigen. And we started out in stage 1, very rapidly producing a cell line at 1 gram per liter, then took it up to 4, then to 10. And now we're getting, in this particular case, 20 grams per liter in 7 days. As I mentioned, the site-specific integration, you can see here, this is an example of what we did during COVID. We took the Wuhan RBD, we put 1 copy in the far left here on the RBD site 1. We could have put 2 copies in there, and then we could have put another 2 copies in the back end where you see RBD2. We did the first step of the RBD within 60 days. We had stable cell line, we had purified protein, and in this particular case, we used a C tag so that it allowed us to switch and without having to go and redevelop a downstream purification all the time. The C tag gives you up to 90–95% purity, and it's only 4 amino acids. It's been used in the malaria vaccine in Jenner, and we use it in our phase 1 successful trial in South Africa. And we're working on a even faster way, you can see here in the bottom, to try to get this done within 35 days to have your master cell bank and your working cell bank ready to go for a pandemic. So one of the advantages of the C1 cell line is not just the hyperproductivity and the speed, but the complexity of the types of proteins it can produce. We've produced VLPs, in this case, Gumburo at 2.24 grams per liter in 7 days. We've produced C1 trimers for the hemagglutinin. We've had H1, H5, H7. You can see on the bottom, we had produced a fusion protein, a MHC2 targeting protein. And then we're going to focus today on what we call the C1 ferritin nanoparticles in a 1-step production. And these are some examples. We did the F10 Wuhan GRBD at 3.5 grams per liter in 5 days, the Omicron at 0.7 grams per liter in 5 days, or the particular Astracon, the one we're talking about today, for the bird flu or pandemic influenza at 2 grams per liter in 7 days. The advantage of C1 also over certain cell lines like yeast, some people use Pichia or Saccharomyces, but the genome of C1 is 4 times larger. So it allows you to make these ferritin nanoparticles we just talked about, but also the full spike protein. So here you can see that we produced a full spike on the initial stage 1 without doing any fermentation optimization at 1 gram per liter in 7 days. And the performance you can see on the right, it was very immunogenic. So from a fed batch fermentation, whether you use stainless steel or single use, the cell line, because of the unique morphology, allows you to use also single use fermentation. And then the downstream processing is very similar to anything because it's a secreted protein. So keep that in mind that this protein is secreted for 95% of the things we do. Fully defined low-cost medium, fed batch technology with glucose feeding. Again, 4 to 7 day fermentation process time, no endotoxins or viral clearance are needed. And again, we're scaling down from 500,000 liters to scales that are being used in the pharmaceutical industry. And we need no inducer and is low-cost chemical defined media. So in terms of speed and advantages, you can see here there's less time, no viral clearance needed again, hyperproductivity. This slide shows you the comparison of CHO cells, which take up to 54 days between freezer to the shake flask and the scale up and the final fermentation process being complete on the upstream. Insect cells like baculovirus cells can take up to 38 days, and C1 typically runs in a 9-day to 12-day fermentation from freezer to shake flask to scale up to having secreted protein with a C-tag purified. An example of the SBV, Schmallenberg antigen, was run in the European Union, the Zoonosis Anticipation Preparative Initiative, ZAPI, independently, where we produced, in this case, was 1,800 milligrams per liter. And by the way, that was at stage 1 without any optimization, versus baculovirus, we got about somewhere in the neighborhood of 300-fold higher productivity or higher yield. So some of the advantages or the disadvantages of the other systems, you need viral clearance for downstream release for both CHO and baculovirus, longer, more costly upstream and downstream processing, 20-hour doubling time versus 2.5 hours for C1. We have a narrow range of temperature and pH. We can go from pH 5 to 8, 20 to 45 degrees Celsius. We can produce 3 batches, the 3 to 4 batches. In the same time, you would produce, excuse me, 1 batch of a baculovirus produced product or CHO cell produced product. And one of the things that's interesting, again, because we produce sometimes VLPs or nanoparticles in very large, in one example, one of our collaborators was producing a nanoparticle vaccine. It was quite large in CHO cells, and it was too large to actually go through the viral clearance. So in the end, they were having trouble with the FDA trying to get an approval because you couldn't viral clear the particular protein. This is a case study of what we did here for the preclinical DYA100 COVID booster vaccine. We used the preclinical studies for the rabbit studies, mice, cattle, lambs, chickens. We've done all kinds of things in preclinical development. In this case, we did the New Zealand white rabbit tox study. We'll see that later. And we were able to demonstrate significant persistency following 42 days. Completed cGMP manufacturing of toxicology and clinical trial material, developed full regulatory process under SAFRA in South Africa, and then fill finish completed, CMC, et cetera, through a successful phase 1 trial where we demonstrated safety, it was well tolerated, and then it also induced robust immune response in both humoral and cell-mediated responses in the vaccinated individuals. So here again is just an example of the purity here on the left. This is the antigen that I showed you earlier, where we started off at 1.1 grams per liter, moved it up to 4.2, and with process optimization, got it to 20 grams per liter. Now, this is obviously an extreme case of high yield, and you can imagine just how low cost and the quantities of vaccines you can make using this particular antigen. Here's an example of just some of the things we made. The Astrakhan that we're talking about for the H5 bird flu, you can see here, was 1,970 milligrams per liter, or 281 milligrams per liter per day. Ferritin nanoparticle, the H1N1, which is the Vietnam, was again 2,000 milligrams per liter in 5 to 7 days. Neuraminidase, antibodies, you can see here a variety of different antigens at very, very high levels. And most of these, other than the one that was 20 grams per liter, these were stage 1 unoptimized. So now I'm just going to go over some of the data on what we have as the cross-protective adjuvanted self-assembling nanoparticle H5 2.3.4.4b subunit vaccine candidate. This has been initially tested in serum for cattle. It's been used in a poultry trial. And so this could be used for animals and human vaccines. And the cost to produce this allows you to do both. And I know in Southeast Asia, there's a lot of cattle, a lot of poultry that's being produced, but this is targeting for a human vaccine, but it can also be used for animals. So again, we talked about fermentation results. Day 6 was 1.5 grams per liter. Day 7 was roughly 2 grams per liter. And then you can see the purification here from the gels. So think about what that means in terms of scalability and affordability. And both of those are very important because, of course, if you don't have access or it's unaffordable, a vaccine or an antibody or treatment is 100% ineffective. So prior to doing any strain and fermentation optimization, as we mentioned, this H5 clade 23334B was 2 grams per liter in 7 days. Using a 2,000 liter fermenter and a standard E. coli microbial bioreactor. So you don't need any fancy equipment. You can run it in a standard E. coli bioreactor. And in this case, we're using example 50 micrograms. Our rabbit study was actually 30 micrograms. So this is conservative with a C-tag to purify it in 2 weeks using chemically defined media at 2,000 liters or 2,000 liter scale. You could produce 40 million doses. If you had a 15,000 liter microbial bioreactor size, and remember, we're going from 500,000 liters down, so scaling at 15,000 liters is something we've done for almost 2.5, 3 decades at this and larger scale. You could produce 300 million doses. So not only could you reduce the cost, you not only get to do it fast, you can produce the large quantities that don't need to have viral clearance or endotoxins for clearing downstream. So you can just imagine the difference between distribution, it's stable at 2 to 8 degrees. So versus mRNA, where you need to obviously have cold storage. I think the cost is a fraction of the cost. It's easier to use, simpler manufacturing process, standard equipment. So C1 is a sustainable and affordable alternative to mRNA and other vaccine processes. So we did a study with a company called ViroVax, and ViroVax, by the way, develops adjuvants for inducing immune response. Worked with the NIH for the last 15 years, funded by the NIH here in the US. We developed what we call this H5 subunit ferritin nanoparticle vaccine. In this particular case, you can see that the immune response and the neutralizing antibodies were really, really through the roof. The median titer was 11,000. So neutralizing antibodies is not the standard way that people, I think, measure the immune response. They usually use what's called hemagglutinin inhibition assay. And this was a serum that ViroVax had sent to the University of Wisconsin to Yoshihiro Kawakoa in his laboratory. And Yoshihiro is one of the world leading H5 influenza experts in the world. And he compared it to his standards, and it just blew it away in terms of hemagglutinin inhibition. So it shows that the hemagglutinin inhibition and the neutralizing antibodies correlate virtually identical. So the next thing that happened here, as the virus evolved and moved into the US into cattle, we then ran a cross-neutralization study with a prime and a boost. And you can see here, we did it with the Astrakhan H5, the A Texas, and then the dairy cattle. And this is the regimen we used. In this case, it was 25 micrograms of the adjuvant and 30 micrograms of the antigen. So this just shows you that all three of the different clades had extremely good protection. In this case, the median with all three was 9,000. Which is again very, very high. And this is the example of the Astrakhan itself. And then this is the example of the H5A Texas. And then this is the other example here, which was the dairy cattle virus. So you can see that the protection is extremely good using this particular candidate, the nanoparticle C1 produced nanoparticle vaccine subunit, with a very powerful adjuvant. So I'm going to go in further on and beyond just strictly using this for vaccines in the appendix and go through monoclonal antibodies, because I know actually some of you have already reached out and said you have FC fusions, you have Fabs, you have monoclonal antibodies, you have other therapeutic proteins. So we worked with Jim Crow's lab during the pandemic, and prior to that, on Andes virus, on Zika, on Rift Valley, and SARS-CoV-2. And you can see that the neutralization and binding is identical to the monoclonal antibodies produced from CHO cells for all these different antibodies. We then did a preclinical study. This particular study was done by Albert Osterhaus and a group of scientists in the European Union. And they first did a hamster study, and then they did a non-human primate study. And it showed that the C1 produced mAb was equal, if not better, than the mAb produced from the CHO cells in solving the problem here for a COVID antibody. This particular antibody also worked from Wuhan all the way through Omicron 2. It could be produced faster, quicker, cheaper, and there's a publication in Nature Communications that we can send you if you'd like, or you can look up on our website under dyadic.com. And then you go to Media Center and Scientific Publications, and you'll find this and several other publications. So here's an example of not only can we produce mAbs or antigens for infectious disease, but for other diseases as well. This is an example of a nivolumab, which is BMS's Opdivo, which is an $8 billion cancer antibody. And here we produced this at 22 grams per liter in 7 days. Again, we had knocked out 15 of the different proteases so that it was quite stable. And we made a humanized version at 9 grams per liter in 7 days. As you can see here on the right, the 99% human-like glycosylation structures after we glycoengineered the C1 cell line for monoclonal antibodies. We worked with the US NIMBLE. It's called the National Institute for Innovation and Manufacturing, where there's, I think, 300 to 400 members from biotech, academia, pharmaceutical companies. And they compared C1 on 2 different things. One was a mAb and one was a Fab, which was the Regeneron Fab. And you can see here from this that their basic conclusion is that these features make C1 an outstanding production platform compared with other recombinant protein systems, such as yeast or mammalian cell platforms. It outperformed it in terms of speed, yield, and cost. From a diagnostic standpoint, as an example, we worked with this is a poster that was presented by UC Davis, and they demonstrated that in a 3-liter fermenter, in one run in 7 days in the fermenter, they were able to generate 120 million doses of an ELISA test. So from a standpoint of a pandemic preparedness, we can produce antigens and antibodies for diagnostic use so that test kits will not run out of the antigen or the antibodies on a global basis. So I just go through quickly here the successful human clinical trial of a C1 GMP protein. Again, I mentioned we ran a phase 1 successful trial, and it showed, by the way, well tolerated and safe for injections in humans. The vaccine induced robust immune responses, including both humoral and cell-mediated responses in the individuals. So we worked in this particular case on the C1 or the SARS-CoV-2 RBD. As we mentioned, in approximately 2 months, we developed a C1 strain expressing the RBD. We also, as I mentioned, have expressed a full spike protein as well. It was stable. We got it purified. It was at about 1 gram per liter when we started. We've actually made it at upwards of 2 grams per liter in 4 to 5 days with little engineering and not a lot of optimization. And you can see the purity on the left through the C tag on the right. So this is a study that was done. I think there was up to almost a dozen different mice studies that were done in different parts of the world using this C1 produced RBD. This was done by the Israeli government. They showed, by the way, prime, a boost, and a booster. Long before that the world thought about a booster, they had already run a 3-dose regimen of the RBD, and you can see very high neutralizing antibodies, both after the prime and boost, and then even higher after the prime and the boost, and then the 3rd shot, or even higher. So we then took this in transgenic mice, ACE2 mice, and showed from a challenge study. We used L-hydrogel 85, 2%. So we used a well-known adjuvant along with the antigen. This was something that we couldn't get our access to some of the more powerful adjuvants during the pandemic. So we used alum. We knew it would be safe. It had been used for decades. And you can see here that it had great protection in challenge studies. We then did a tox study where in this particular case, we actually used 4 different shots a week apart, and it showed great performance in terms of both efficacy and safety. And this is also on our website, the full tox study. You can look that up as well. And most importantly, we had the successful DYA-100 phase 1 results. It met the primary endpoint of safety and tolerability. And again, it induced robust immune responses. And these are the different patients. You can see here that there were no adverse effects. And I think it was probably more tolerated with less pain and less effect, I think, than most of the other vaccines that were tested. We then produced not only the Wuhan, we produced Omicron 1, Alpha, Beta, Gamma, Delta, BA5. So you can see here that we can very rapidly swap out the gene of interest without having to go back and start all over. So from a regulatory perspective, it's beautiful because if you use the same adjuvant, you could pop out the one gene, put the next one in, and move on to the next variant of concern. So we could very rapidly not only produce stable cell lines, but we're talking about producing stable cell lines that produce antigens in global quantities at a cost that provide access, affordability, again, 2 to 8 degrees storage. And I think it's a great solution for any country and certainly for Southeast Asia. And we've also put in this particular example, alpha, beta, and gamma. We produced all three of those, could have done alpha, beta, gamma, and delta, but at the time there was no delta. So we actually did alpha, beta, and gamma all in the same cell at the same time. Today, we could even put in more using CRISPR. This was using the strategy of the site-specific integration, where we can put 2 copies in the front, 2 copies in the back, so 4 copies. With CRISPR, we can put in 5 or 7 different copies, and you can actually have a multivariant in the same cell producing at the same time in a fermenter rather than individual, let's say, fermentations and then blending. It would be a more efficient, quicker, better way to do that. From a regulatory perspective, if you can have consistency from batch to batch to batch of your multi-cocktail, it would be a wonderful thing. And here's just an example again, whether it's an antibody or an antigen, we can blend them individually, or we can make them in a multivariant cell line. So C1 is a very versatile protein production platform. So one of the benefits of C1 is it can be used to make a lot of different things. And so one of the problems we have for pandemic preparedness is people build these facilities and then they mothball them because there's nothing to run in them. In the case of C1, you can scale down because of the yield. So for example, instead of spending $1 billion on an mRNA facility in Kenya and then having Moderna walk away, you can have small footprints. For example, you could have a 100-liter fermenter, 2,000-liter fermenter, 50-liter fermenter, whatever you want from a size standpoint. You could produce complex subunit vaccines, whether it's a full spike, a ferritin nanoparticle. Neither one of those are producible in yeast, for example. The genome of C1 is 4 times larger than yeast. It folds better. It has better complexities in terms of being able to make disulfide bridges. You could produce different antigens, multivalent RBDs for animal and human health. You could produce monoclonal antibodies, again, for animal and human health and for a variety of other conditions. Or you could produce things like insulin, potentially, and diagnostics. And we have made now human serum albumin and bovine serum albumin at very high levels. We're producing bovine serum albumin at about 30 grams per liter in 7 days and about 20 grams per liter of HSA. We can make certain enzyme catalysts like DNase-1 we're producing and hyaluronidase. So in the end, you have a very, very rapid, more productive, more affordable way to make proteins. You have a lower cost solution. You have no endotoxins or viral inactivation that's required. It's a shorter, faster cycle time. And then the potential to produce more product per batch in larger overall quantities at much lower cost. And with that, I would say thank you, and I'm here to answer any questions you might have.
All right, thank you very much, Mark, for the very nice presentation and very comprehensive as well. And are there any questions from the audience? And if you have any question, you can actually unmute yourself. And I can see that we have the raise up from Hatta. Hatta, please go ahead.
Okay. All right. Good morning, Mark, and good morning, Professor Wong, UK time. Okay. I'm Harta, a fresh graduate clinical microbiologist from Bali, Indonesia. So with this kind of vaccine strategies, is it possible to develop or manufacture vaccine for dengue fever, Mark? Because we are currently in Indonesia, we have from Den 1 until Den 4 for the dengue fever virus. And is it any possible way to manufacture the cocktail virus composition for the dengue fever virus? Thank you.
Yeah, no, I mean, again, as I mentioned, we're working with academia, industry, and government agencies, and also obviously nonprofits all over the world. We worked in South Africa, we worked in India. We're in discussions with establishing a manufacturing facility based on our technology in Bangladesh. We're happy to work with you in Malaysia and anywhere else in terms of enabling you in two different ways. One is if you have gene sequences that encode for an antigen, for example, whether it's dengue in this particular case or something else, we can develop that stable cell line for you very quickly that has high productivity and very low cost, and then transfer the technology to you to produce in an E. coli fermenter of virtually any size imaginable. So that's one way of doing it. The other way is we could license you the technology, like what we call the full license kit. And in that case, you would get all the genetic tools to do all that work yourself on site. And we would train you how to take genes, insert them into the genome and the chromosome in either a site-specific manner or a random, or if you have a license for CRISPR, you could use CRISPR. So there's multiple ways of doing transformation, and then we would basically tech transfer the whole platform to you to be able to use it to produce a variety of different things for different applications, whether it be animal or human health, and or other products from recombinant proteins. So those are the two different approaches that we've used. And I think one of the things that we suggest is that until you become proficient, you let us make the cell lines for you. Because if we do it, we've been doing it for 30 years, we're going to get a higher cell line productivity. And of course, if we can get something at 2 or 5 grams per liter and you get it at 300 milligrams per liter, using the cell line we generated for you may cost you 20, 30,000 euros more for the cell line, but you save production for years, for 20 years at a much higher cell line. So you can save hundreds of millions and billions of dollars by making sure that you have the most productive cell line at the beginning. But you could also do it on your own and ultimately, hopefully get similar results to what we do with our experience. Did that answer your question?
All right. So it elicits me to add one more question. Can I?
Sure.
Yeah. All right. So with that, I see there is any possibility for manufacture a vaccine for multi-drug resistance bacteria, Mark, because in hospital setting, especially, it will become the silent pandemic in Indonesia, especially in the tertiary hospital, the MDR bacteria or MDR microorganism that causing the terminal diseases for the patients.
Right. So again, excuse me, I actually have a virus I caught somewhere that I'm recovering from. We can make proteins. So if you have a gene sequence that encodes for a protein of interest or a target protein that can address that problem, we can develop a cell line to produce it. We also work with scientists all over the world, including, you guys may know, Rino Rappioli. Rino Rappioli is one of the most renowned vaccinologists, scientists. He just retired from GSK about a year and a half ago and started a foundation, Biotecnopolo at Siena in Italy, and has a pandemic preparedness center. And he's working with us with the C1 cell line because he recognizes there's a gaping hole in having the ability to produce access and affordability for countries around the world. So again, we're agnostic as to the protein you want to produce. The complexity and the larger genome of C1, filamentous fungi, that doesn't sporulate, that has a low viscosity, that can run under pH 5 to 8 or temperatures of 20 to 45 degrees. It gives you greater flexibility, robustness, and versatility in making recombinant proteins. So if you have a sequence, we can develop a cell line to produce it. And if you don't have a sequence, we can work together and try to identify something that may solve that problem.
All right. Thank you. I got it.
Great. Thank you.
Yeah, thank you very much. And any more questions from the audience? Kang Lan? Kang Lan, please go ahead with a question. Yeah.
Thank you. Sorry, I couldn't unmute myself. Thanks, Mark, for the presentation. It's very interesting. I'm Kang from the University of Sheffield. So I'm quite curious about, because one of the things you mentioned quite early on is the change in the morphology of the C1. And are you able to comment on how this change was brought about? How was it achieved? And also, how key was that in the improvement in the productivity of protein?
Yeah, you can see on this slide on the left here, and I can probably pull it in a full slide deck again. Let's just see here. See if that works. Is it coming up? It's not coming up now.
I think it's at the end of the slideshow.
Yeah, no, I went back and put it back up.
Ah, okay.
I think, I don't know if you guys have been seeing it, but anyway, they have a slide here. Can you see the slide?
Yeah, I can see the slide.
So here on the left, you can see morphology, which as you see, filamentous fungi typically grow in very long filaments. And we changed the morphology. It was done by random mutagenesis early on through UV mutagenesis and then chemical mutagenesis. So this was a kind of an aha moment, okay, serendipity that just happened. And so we discovered it. And by the way, it starts out in the filamentous fungal form, and we know how to, there's like a switch in how we run the fermentation that changes that morphology. So it's very unique and novel. And yes, it ends up with more tips. And filamentous fungi secrete proteins from the tips. So you think about it, if I took a hair out of your head and stretched it from the left to the right, you'd have two tips. We took it and made it into whiskers. So you have hundreds of tips and the same mass secreting protein out. So it led to hyperproductivity and it led to scalability, low viscosity, uniform feeding of nutrients and aeration. So it was a major change in that regard. And then we didn't stop there. Then we had to knock out all these different proteases because, of course, filamentous fungi and other organisms produce proteases that'll chew down an antigen or an antibody. And we have 15 proteases knocked out that still provide productivity without killing the cell, letting it still grow in a healthy way. And so we have different versions from a 3x, from a 4x, a 9x, and a 15x. So depending on your antigen, if it can be run at a 9x protease knockout, you'll get even better productivity than a 15x. But to be honest, our productivity is world-class anyways and far above anything else that we typically just use the 15x for a recombinant protein antigen or an antibody to produce high levels of very stable proteins.
Okay, so is the current C1 strain a strain whereby it's constantly in the slightly grain-like morphology, or are you controlling the morphology based on the fermentation condition?
Well, no, we're controlling it based on the fermentation conditions.
Okay, I see.
But the cell line, the predecessors would not be controllable that way. So you would have to have this particular mutant, like the wild type strain, the first generation, second, third, would not have this morphology.
I see. Just out of curiosity, how long did it take you guys to change or to actually enable this functionality?
Over several years.
Thank you very much.
All right, thank you, Kang Lan. And any more questions from the audience? I have actually, oh yeah, Nanny, please go ahead, yeah.
Hello, hi. Can you hear me?
Yes.
Hello, Prof. Tak. And hello, Mark. Thank you for your interesting presentation and your C1 platform. I'm Neni from BioPharma. So what I would like to ask is, you mentioned about C-tag protein. So is the purification involve the affinity to the C-tag protein, or do you have any kind of the purification process from polishing to the intermediate polishing to the polishing? And how you test the residual host protein of this C1? Are you provide the kit for this residual protein? That's the question. Thank you.
Yeah, so the CTAG is a Thermo Fisher technology that we use. We use it because it's only 4 amino acids. It's been used by the Jenner Institute for their malaria vaccine. We've used it in our phase 1 clinical trial. We obviously made the C1 RBD after the CTAG and purified to get 99.4% purity. So unlike insect cells, we don't have all these issues in regards to purification. It's a simple process. It's a secreted protein. And yes, Cygnus, which is one of the leading people for kits for host cell proteins, has worked with us, and they have a commercial kit for C1 that you could purchase. C1 whole ACP kit.
Okay, thank you. Another question is, do you have any problem with glycosylation to specific monoclonal antibodies that especially in the FC region, some of these monoclonal antibodies need specific glycosylation pattern?
Right, so we have different glycosylation cell lines. So we've engineered C1 where we have G2, G1, and G0. We actually have cell lines which has no MAN5, because of course in CHO cells, MAN5 over 5% is a problem. So we don't even have that. We don't have fucose. So for example, for neutralizing binding antibodies, you don't need fucose. Our ADCC is much higher. So in some ways it may actually perform better for cancer or some other areas. We are now having some discussions with people who are looking for our nanoparticle vaccine for cancer. For example, some of these mRNA vaccines that are being used with Keytruda as a combination therapy, you need 9 or 11 shots. It's almost insane if you think about that, but you know, with a ferritin nanoparticle C1 produced, we think that maybe that same process can have 1 or 2 shots and get long-lasting durability. So, you know, we think that it's critically important, and I think the main message here is when you start your development process in preclinical studies, typically you take what you have on the shelf. You take E. coli, you take yeast, you take CHO cells, you take insect cells, and you built in an inefficient manufacturing process. And you don't find that out until you get into phase 1, and then all of a sudden you can't go back. And what happens? You end up with it, if you're lucky and the product works and you get to phase 3 and you commercialize it, you fall short in access that's too high priced. You limit the audience, and then the world doesn't get treatments. So health equity is a solution that I think is achievable if we actually pay attention at the beginning and use more efficient cell lines that ultimately can produce more for less faster, and that we can treat more people. And so big pharma sometimes doesn't pay attention to that. Academics certainly don't pay attention to that. And we build this inefficient cycle of, I think, it's just almost mind-boggling when you think about it. But we need to get academics and industry to think about it. If it takes you a week, a month, 2 months longer, other than a pandemic, which we can react to, you can see very quickly anyways, you're much better off to use a more productive cell line, because then you can produce more for less. You can run 1 fermenter instead of 10 fermenters. You can get a higher yield, a lower cost. You can increase access and affordability. And this whole health equity situation can be solved to a great degree. Now, monoclonal antibodies is a great example. During COVID and during Delta, with all the world's resources thrown at it, we couldn't make enough antibodies for the USA, let alone anywhere else in the world. That's just making them at any price. So the bird flu is 30% deadly. That's a 1% deadly disease. If we have a serious outbreak with a deadly disease or lethal disease, a lot of the population in the world is not going to be here. And CHO cells are just not going to cut it. We're not saying get rid of CHO cells. We're saying let us supplement CHO cells and have the ability to make larger quantities more affordable so everybody has access. So we really need to think about it. In the monoclonal antibody space, to your point, it's the gaping hole of health equity is you drive a semi through it. It's just a massive hole that needs to be filled. And our preclinical study in the non-human primate shows safety and efficacy on par to CHO cells, but faster, larger volumes, and lower cost.
Mm. Yeah. Can I ask one more question, Prof?
Yeah, definitely. Go ahead, Nanny.
Yes. Because in Indonesia we have a halal regulation. So I would like to ask you about the medium and the process. It is involves any animal origin, or you are using non-animal origin medium and material. Thank you.
Yeah, so to answer your question, it could be halal certified. We used to make industrial enzymes for food and feed, and they were halal. So that's a very good question, and I should have brought that up on my own. But yes, unlike some of the other technologies, we completely can be halal certified.
Good, good. Thank you very much, Mark. Thank you, Prof. Tak.
Thank you very much. And any more other questions from the audience? Yes, please go ahead, Aria.
Okay, thank you so much, Prof. Tak and Mark, for your time. I am Arya. I am currently as a student in the University of Sheffield, and I'm also working in biopharma. It's a very interesting platform for me since this is the new as well for me. Regarding the glycosylation, I saw from your presentation that the glycosylation pathway has been engineered. Have you assessed the genetic stability of this particular strain or platform? And what is the result? Because you know that the human-like glycosylation is very important. That was my first question. Thank you.
Yeah. Excuse me. So we do have a human-like cell line where we've glycoengineered the cell line. And then we have what we call is a human-like cell line, which is the man 3 to man 9, typical, what we would call the C1, which is more human-like than typical fungi or yeast. So the actual non-human primate study was conducted with a non-human glycan, and it was shown to be safe, effective, and protective with no ADE. So depending on what you're doing, if you're doing a one-shot, sort of like somebody's in a hospital, they got COVID or they got some kind of viral infection and they're going to get treated once, you don't need to have the human-like glycans, the identical glycans. It neutralizes and binds identical. If you remember, I showed you the different antibodies from Jim Crow's lab, whether it was Andes or Zika or COVID. And we've seen that time and time again, that the binding and neutralization is identical. For a repeated dose, to your point, people think about you need mammalian-like structures. I mean, CHO cells are not human, CHO cells are hamsters. So the point is we've made a human-like glycan structure for those applications. So we have both sides. For a pandemic, when you're going to need massive quantities at low cost, I would suggest you use the non-human, but the human-like, because the more people you can save, the better at a lower cost. And so the productivity is about twice as productive in the human-like natural structure as it is in a humanized version. But we have both.
Okay, got it. So you have both in between the human-like glycosylation and the other one. Okay,
Right.
and actually I have one more question regarding the sporulation. And you have mentioned that this platform doesn't sporulate. Is there any test method that can test there is no sporulation, especially where we use the machine or equipment to produce this platform? Because in the regulation, we often ask about the cleaning validation, especially if this platform is sporulate, and you say that this platform doesn't sporulate. Is that right?
Yeah, it's a non-sporulating filamentous fungi. Typical filamentous fungi do sporulate. We produce on an annual basis in our R&D lab, just as an example, 300 to 400 fermentations. In the same fermenters, we use yeast and other things at the same time. And so we don't have sporulation with C1. I think that has something to do with the morphology as well as the genetics. So we did run under CGMP in a standard E. coli fermenter in a facility that normally runs E. coli. So they ran several batches of C1, and then they ran E. coli in the same fermenters later and prior to that. And of course, there's cleaning methods. People think about sporulation like bacillus. You know, even if it's sporulated in predecessor strains way back, the spores are completely controllable in that regard anyways. It's not like people think in a typical like spreading of like a bacillus spore. So even if you had it, which you're not going to, you can clean it up. And so it's been run under CGMP not only in Israel, it's been run in India, where they ran and they did the air filtration and the standard production for CGMP, and showed there's no spores in the air from C1. There were plenty of spores, but not from C1.
Yeah. Okay, I see. Because sometimes our regulatory bodies asking about the cleaning validation, and since this technology is quite new, maybe for us in our country, maybe they know that the fungi is normally spore related, but sometimes we have to find our way to explain them that this strain doesn't spore relate. So yeah, that's kind of a good idea. Thank you for your help and your answer, Mark. Thank you so much.
Yeah, no problem. Thank you.
Indra, please go ahead.
Hi, Mark. It's really a nice presentation. Really appreciate that. I just have a couple of questions related to the regulations. Do you have any visibility on the because this platform is quite new, and also it's quite novel technology as well. Do you have any visibilities on how the regulatory body will accept this technology? And the second one is, do you have any also kind of like a strategic plan to scaling up the production using this platform? Thank you so much.
Yeah, so as you see from the slide, we have actually received US FDA GRAS status, where we actually in the industrial space injected the enzyme without purification in rats and mice, and actually there were no toxicology issues at all, and so we have a GRAS status. But we've also run a phase 1 clinical trial now in humans in South Africa with SAFRA, which is a regulatory agency, the South Africa Regulatory Agency. Of course, we are in discussions with the US FDA. We worked with the Paul Ehrlich Institute, or the PI, in Germany for our preclinical studies and to get ready for the IMPD for the filing. So I think the regulatory agencies, that we've spoken to, have basically said, look, show us the data. If you have safety data in your preclinical studies, and we don't care what you use. If you use a tobacco plant, you use a yeast, a fungi, a bacteria, or whatever, a CHO cell. If you have safety data and efficacy data, they're going to accept the platform.
I think there's a second part of the questions related to scaling up.
Oh, okay. Well, we're scaling down. So typical biotech has failed to scale. Remember, we've run this for over 2.5 decades at 300,000 liters, 500,000 liters, 150,000 liters. So scaling it up, we don't have the same problems we have with CHO, where you start out at 1 liter, you go to 10 liters, you go to 100 liters, you go to 1,000 liters. We went from 5 or 10 liters to 120,000 liters in scale up. So in this particular case, we're actually scaling down.
Thank you so much, Mark. Really appreciate that.
Yeah, thank you.
Any more questions from the audience? We have 4 more minutes. Okay, I have 2 very short questions for Mark. And you mentioned that the protein is actually secreted by the organism. So do you actually know the size limits of the protein that can be secreted?
Well, I don't know. I don't have the limit, but we make full-length monoclonal antibodies, and we made the full spike protein, and we make nanoparticles. So, I mean, obviously, we've produced things at 150, 200 kilodeltas, but, we've never pushed the limit. But so far, I think we've been able to produce virtually everything we put into it in terms of sometimes in a VLP, for example, we get a secreted protein, and sometimes it's intercellular. It just depends.
Okay.
It depends on the gene sequence.
All right. So have you actually seen any correlation between the productivity and the size of the protein? Is there any correlation?
I mean, there's obviously correlation because when you have a smaller protein, obviously you're going to get less grams per liter.
Okay.
But we've got 20 grams per liter of that antigen for the animal health, which is a very small protein, right? For
Yeah.
the antigen, and then a monoclonal antibody, we've had 20 some grams per liter. We made an enzyme at 80 grams per liter. So
Okay.
I think the issue here is CHO cells have been used for 35 years, media optimization, billions and billions and billions of dollars. We're just getting started and we're already outperforming them in speed and yield and cost.
All right, thank you very much. And my one last question is related to the carbon feedstock. You actually mentioned just now that you actually use synthetic media. So how flexible is this organism in terms of taking other carbon feedstock, for example, like lignocellulosic material? Let's say we want to produce a enzyme for industrial application. We want to actually utilize a cheaper carbon feedstock. Have you actually explored how versatile this organism is in terms of taking up carbon from other sources?
Yeah, I mean, if you remember, we actually come from the industrial enzyme
Yeah.
space. So, we in that case, it wouldn't be chemically defined media, but we've used the waste from a sugarcane bagasse,
All
like
right.
molasses, we've used sucrose, we've used glucose. So it can grow on all kinds of media.
All right, thank you so much. Yeah. So, we are running out of time now. And thank you very much for joining the seminar. And thank you very much, Mark, for the very, very exciting talk. I'm sure that we have learned a lot from Mark today. And so if we have permission from Mark, we will actually share your contact details with all the participants as well, in case they would like to actually contact you in the near future for collaboration, because I know that your technology is actually very exciting and it definitely has a lot of application in terms of production of halal compliance vaccine, for example. And I hope that this is actually a good starting point to actually create more communication and also connection, interaction in the near future. And thank you very much, everybody, for joining the seminar today.
Thank you.
Thank you. Bye,
Thank
Mark.
you.
Thanks a lot. Thank you.
Professor.
Okay.
Thank you,
Bye-bye.
everyone.
Thank you. Bye.
Bye.
You.